What are random mutations and why are they such a great deal in the theory of evolution? Today we'll understand in very simple language about genetics, replication, and random mutations so that we can follow through later about what is the role of random mutation in the theory of evolution. So let's explore. Welcome to this episode about random mutations. The theory of evolution is about the change in features, traits, and physical appearance of organisms over a long time. But the theory of evolution also claims that those changes, the big ones of those changes that causes the creature to become apparently a completely different one to an observer, are due to a series of accidental events, random mutations that are acted upon by natural selection. The physical appearance of an organism is called its phenotype. It is the expression of the genetic material of this organism. Genetic material codes for those phenotypes. And the collection of this genetic material, of those traits in the genes, is an individual's genotype. But how do those genes affect the external appearance? How do genotypes affect phenotypes? Well, we need to understand a little more about genetics first. Every one of us has inside him a book of instructions that we call the genetic code or the genome. And like human books, it is made up of letters. But the letters of the genetic code are only 4 letters. They can be A, T, C, or G. So you have a big series of letters forming the genome. Letters come together into sets of 3 that are called codons. So a codon will have 3 letters, one after each other. Each one can be an A, a T, a C, or a G. So each letter can be one of 4, and you have 3 of them, which gives you 64 different combinations. Each combination, which is a codon, is like a word in our book. But those words, some of them have similar meaning. So those 64 words do not give 64 different meanings each. No. 61 of them have synonyms, and when you take, for example, these 6 codons have the same meaning, so you count them as 1, the 61 will correspond to 20 codons that code for amino acids. And when you produce amino acids and arrange them in a chain, you form a protein. The 3 other codons code for a stop signal. It's like a full stop at the end of a sentence. So we have the letters, which are also called the nucleotides, and we have the codons, which correspond to the words, and codons arranged after each other give us sentences or paragraphs. And those paragraphs will convey, like a paragraph in a book, a certain something that the book is saying, an instruction for something. Those paragraphs are we know as genes. And genes can be, for example, an instruction to manufacture a protein. And proteins are very important because those proteins which come into strings of the 20 amino acids are then folded into a 3D shape that performs a function. For example, 2 of those proteins come together to form the hemoglobin molecule, which carries oxygen from our lungs to the cells of our body and is part of our breathing process. Now, those genes are not sitting in isolation. They are arranged together in a string form, one gene after the other, and there might be some genetic material in between. And this long string of genes is corresponding, for example, to a chapter in our book. And this chapter of genes is what we call a chromosome. Now, we don't just have one chromosome, but we have many chromosomes. For example, the human being has 46 chromosomes, 23 coming from his mother, 23 coming from his father. And the collection of his chromosomes is the human genome. Now, within the human genome, the chromosomes are arranged into this fibrous structure called chromatin. And there are those other proteins sitting around, like histones, that can regulate those genes. So a histone modification will cause a certain set of genes to be less expressed. You don't need them. The body does not need them now, or becoming less accessible, and hence they are not manifested. There are other kinds of punctuations that can take place to the genes through something called methylation, for example. And it's like reading a paragraph with a comma in a different place. So maybe this paragraph, with changing the commas, can give a different meaning. So one gene can have different manifestations. The book of instruction also has stories that have specific contexts. So, for example, when a sperm comes together with an egg to form a zygote, and now we need to form an embryo that would develop into a fetus that would become a full-grown human being, we need to understand, or the system needs to understand, the story of how to build a body. So there will be part of the genome called developmental gene regulatory networks that will have this story in a program-like format, in a hierarchical manner. So it says, okay, the body is, has a center axis that is like a mirror, left and right. It has a head, a torso, and limbs. And then the head goes to the level of the head, has eyes and ears and nose. And then the eyes would have a retina and a cornea and a fluid and etc. And then the retina has rods and cones, and then the cones is made this way. And all of this is arranged in a hierarchical set of instructions that are coded into the genome and will direct this part of the story, will tell you, go and read the paragraph about how to create rods and cones and create them. Go and read the paragraph about how to create neurons and create them in the brain, etc. So those instructions will regulate and will organize how the other genes in the other chapters of the book will produce, for example, the body during embryonic development. And there are other types of gene regulatory networks. So starting from the very first letters, into the codons that give us the words, into those paragraphs that can be punctuated, into those chapters, into those stories, we have the book of each organism, magnificently written in a way that such set of instructions will become the creatures that we see around us, each of them carrying its own book in each cell of its body, in the case of eukaryotes, where the majority of cell types will have inside its nucleus a complete copy of the book. In mammals, for example, the genome of a mammal is around 3 billion base pairs, 3 billion letters writing down the story that is the book, the instruction book of this creature. Now, we said that mutations are random changes to the genetic code, or essentially errors during genetic code replication. So let's first understand how the genetic code looks like and why we need replication. The genetic code is arranged into this double helix that is actually two strands attached to each other. And when you have a letter on this strand, it is attached to a complementary letter on the other side. It's not the exact same letter, it is a complement. So the A, for example, would bind with a T, etc. So each of the four letters has its complementary letter that it binds to. And so the two are called matching to each other. Now, why do we need replication? Well, because a cell does not remain a cell. A cell would divide, for example, for growth, for example, to replace damaged or dead cells, and obviously for reproduction. So when a cell is dividing, copies of the genetic material are made. So instead of those two helically wrapped strands of DNA, they are taken apart, each one of them is replicated, and then two new double helix strands are formed. Now also in reproduction, the body will copy the genetic material into sperms and eggs, collectively known as gametes, but only one of each pair of chromosomes would go to the gamete. So we'll have 23 chromosomes coming from the male in the sperms and 23 chromosomes coming from the female in the egg. And when they meet, they would recombine into the 46 chromosomes. And then cell division happens under the regulation of developmental gene regulatory networks to form a fetus, which becomes a human being. So the replication of the genetic material is a critical process for the survival of a full-grown creature and for the production of new copies of the creature. Actually, of new offsprings of the creature that are not even identical copies due to inheritance and recombination. So let's look into what happens when genetic material is copied. What you see in the video is this wonderful and amazing machine that's like a turbine that is splitting apart the two strands of the DNA, and then other machines, this one is called helicase, and then other machines are bringing in nucleotides, base pairs, from the fluid in the cell and creating a matching strand versus each strand, and at the end each strand has a matching strand, and you get away with 4 new strands that become 2 new helixes. And this process is so intricate and is so magnificently made that while you are in an environment with all kinds of chemicals, the copying process succeeds in making the correct match. For every 100,000 to 1 million, there is just one error. One error. But still, one is amazed to see that this system does not only copy, but it looks for errors and it corrects errors. So when you see this, you understand that the objective of the system is to make a faithful copy of the genes. And this is why mutations are typically labeled as errors, because the process intends to make a faithful copy. And that's why it has error correction. It has not only one mechanism of error correction, but many. So I will tell you about 3. So first, while it's doing the copy, it has something called proofreading. So it has this letter, and it plugs an enzyme called polymerase, DNA polymerase, will plug the matching letter in front of it. And after it does that, it will proofread. It will read again the letter and make sure that the right letter has come in front of it. And if the new letter is not the right one, it will correct what it has done. This is how this process is designed. Not only that, so now most of the errors that have slipped the direct replication process, the first one, has been corrected by the proofreading. But maybe some of the errors would also slip, or new errors would happen to other reasons. So there is another process that starts after the new strand has been created. A process will match the new strand to the old strand, to the original one. And if it finds an error, it will cut the part of the new DNA that has been replicated. It will throw this part away. It will create a new matching part, and then an enzyme will pass, ligase, that will join the parts together and seal it. So, what is this? This is a process that is so intelligent. It knows what it's doing. It knows it wants to create replicas. It proofreads what it writes, and then it does a review as if you're answering an exam. You might ask, how does it know which of the two strands is the original and which one is the replica? Well, get amazed. The old strand, the original one, has methylation on it. Remember when we talked about epigenetics? The new one did not get its methylation yet. It is going to get it, but it didn't get it yet. And through this time gap, it will recognize the one that has the methylation as the source and the one that doesn't as the copy, and it matches the copy and makes it a faithful match to the source. Now, you have a cell after the division is complete, and this cell is exposed to a chemical external factor or to radiation or something, and one of its letters is changed or is damaged. There is a process that is monitoring the DNA genetic material, and it will go, and when it finds a damage, it will recognize the damage, it will fix the damage by throwing away the damaged part, creating a clean copy, and then gluing it together and sealing it back. So this is repair. All of those layers upon layers are there to keep the DNA material intact. But still, some errors slip. Now, when DNA errors occur, they are now called mutations. And since those mutations are completely unpredictable, they are also called random mutations, because they are due to so many factors. There's a chemical noise, a chemical party happening inside the cell, and it is a process that is, as part of this world, not perfect. So those mutations, when they occur, they can be harmful. For example, this hemoglobin molecule that we talked about, just one error in its chain of proteins will cause a disease called sickle cell disease. This one error, and it has a chain, it's two molecules, together they have 280 plus amino acids. Just one of them is changed, and it gives us the sickle cell disease that causes a change in the folding structure of in the 3D structure of hemoglobin, that causes it to stick to each other and heavily reduces the efficiency of it to bear oxygen, and also affects the shape of the red blood cells. So yes, genes that have random mutations will have predominantly a bad effect, and hence it's called a deleterious mutation. And those deleterious mutations will cause the offspring to be diseased or deformed. And hence, it will have less fitness. Maybe the sperms or the gametes or the zygote or the embryo will die and not complete its development. Or maybe it will produce a crippled or deceased individual that will have less chances to reproduce, and hence is taken, his deformed or degenerate genes are taken outside of the population due to natural selection. Now the mutations that are left, if we put that the majority of mutations, of deleterious mutations, are ruled out by natural selection. The ones that remain, some of them, or the majority of them that remain, that are not ruled out by natural selection, are called neutral. Remember when we were talking about the codons, and that many codons will have the same meaning in terms of producing the same amino acids, and they are hence called synonymous. So natural selection has nothing to say there, because the gene with the mutation that has caused a synonymous codon to exist will produce the same protein, and hence will have no functional change to what the gene does, and hence natural selection has nothing to say about it, and then it just leaves it to be. Now, the theory of evolution claims that there is also a third category of mutations, which are called beneficial mutations, that cause changes to the genes that are not synonymous but has a change in function, and that this change in function will not be deleterious but will give the individual an advantage. Now we need to sit back and think. How probable is it to produce an advantage through random mutation? Well, we need to do some statistical analysis. So here's something, and I'm going to attach a reference in the description. If you have a typical gene that has, for example, 150 codons, that produces a protein that has 150 amino acids. For each one string structure, one peptide structure of this protein, that will deliver a protein that has a beneficial function, that can fold in a 3D shape, that has a beneficial structure, you have 10 to the power 77 other possibilities that will not fold into anything beneficial. Now let's appreciate what 10 to the power 77 is. We are looking for the one that has meaning from 10 to the power 77 arrangements that do not have meaning, until we are able to find the next one that has meaning. There are other estimates, but all of them are ridiculous figures in this order of ridiculity. Let's have a feeling for the number. There is an estimate for the number of grains of sand on planet Earth, that we have 7 multiplied by 10 to the power 18 grains of sand. 10 to the power 77 makes 10 to the power 18 nothing. Because 10 to the power 18, you have a million, which is 6, a million million, which is 12, a million 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 is 10 to the power 18. And then each million you add 6. So 10 to the power 77 is how many million, 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 the number of grains of sands we have on the planet Earth. So with a process that is such extremely difficult, extremely improbable to find the next beneficial protein from an existing one through a random change, we need to sit back and think and look at this system and ask ourselves those questions. Number one, how did such an intricate and magnificent system that does the DNA replication this way come about through a process of random mutation? The system itself needs to be developed from a process of random mutation, because the Origins of Life's project does not claim that DNA was there from the beginning. It was claimed to come maybe from just RNA. So we had a process based on random mutations that started maybe from RNA, according to the Origins projects or theories, to cells that have DNA and DNA replication and error correction. A random process of error generation, can it produce this system of copying and error correction? And then those errors that slip, can they really find the needle in the haystack to produce one protein that has not existed before, that is giving an advantage to a creature that already exists? And even, what about creating completely new genes that give completely new functions? Because according to the theory, every single organism, including the beanstalk and the whale, both go to the same common ancestors, and obviously those two have genes that are not completely novel to each other. So how is novelty arising? Also through the random mutations and natural selection? Well, we leave that for you to think. To give me your questions, if you're not registered, of course, you need to register to the course, and then to send the questions and your ideas to be answered on our live stream coming up next Saturday. Please share this course with those whom you love, and see you on Saturday for the live stream.